Well, good afternoon, everyone. This is Kelly Gillespie. I'm with the Missouri Biotechnology Association, also known as Mobio. We've got several of our members and many of Ed's network friends for our meeting today. We call this Mobile Breakthrough, and we try and do this every few weeks to talk about really some unique opportunities and some unique technologies that are have parts of their founding or development here from the Midwest, and mainly just want to put good people together and try and help that collaboration, that effort that's necessary when we all realize that no one person is going to be a superhero and take it all the way from start to finish appropriately, that it's absolutely a team effort. And Ed Timm is a longtime friend of Mobio, longtime participant, speaker, has helped us on legislative matters, has helped explain science to policymakers, and has continued to make a good living and make a big difference for patients in his career. And Ed, will you please tell us about the new endeavor and the message you want to get across today?
Certainly. Kelly, first off, thanks for the invitation. Secondly, Mobius Otologics is an entity that was spun out of a company that is operating at this time named Mobius Therapeutics. I'm the CEO of Mobius Therapeutics. I'm the managing member of Mobius Otologics. Mobius Otologics is based upon a very unique and very disruptive technology, which we call SPDM, Selective Photodynamic Ergometry. So the entity was spun out of Mobius Therapeutics as a condition of the seed round funding. And this was then distributed pro rata to the seed round investors. The idea being, candidly, you could get, instead of getting one exit, you could get two. And it is a Delaware LLC. We do have issued patents in the USA and around the world. Mobius Therapeutics is an operating entity and is in ophthalmology. Obviously, Mobius Otologics is in ED. The entity status is that it is dormant. And what I mean by that is that we are current. We have been paying our franchise taxes to Delaware. We've kept our patent. We've prosecuted our patent portfolio, maintained the patent portfolio, filed tax returns, et cetera. So from a legal perspective, the entity is still active and alive. However, nothing is really happening as a result of the fact that at the time of the preferred round funding of Mobius Therapeutics, my employment agreement prohibited my involvement in this or in anything outside of Mobius Therapeutics. It's been self-funded by my wife and I since 2009. The NIH, specifically NIDCD, National Institute on Deafness and Communications Disorder, awarded it an SBIR grant in 2009. But as I was the PI on the study, but would not actually be doing the research, the grant had to be withdrawn. I can assure you, though, I know that NIDCD still wants to see this funding. They would like to give us a grant. Proof of concept has been established, and some of the most difficult parts of the supply chain have been established. And that's when you're talking about a technology that actually is in some ways very complicated, having that supply chain established and validated is a huge step forward. So the problem is acute otitis media. Many on this call may have had their own problems with ear infections as a child. Many on this call probably have children that have had problems with ear infection. It's called acute otitis media. And it is the single largest reason a child visits the doctor. There's over 30 million patient visits per year in the USA alone. There's 2 million tubes placed each year in the United States, making it the most common surgery performed on children under the age of 16 years old. And that number is actually growing. Why is it growing? It's growing because what I believe is a ticking bomb in terms of public health, and that is common resistance to common antibiotics. So the bilateral, this is treated ultimately when they go to tubes. It's treated by a procedure called BMT, bilateral myringotomy with tubes. And because the patient is young and because they're non-compliant, they have to do it in the operating room. It requires a general anesthetic. So here you can see an image too of a normal tympanic membrane and then the inflamed tympanic membrane. And behind that, of course, is the fluid. It's important to point out, and this is very critical, the purpose of the BMT is not to place a tube. The purpose of the BMT is to ventilate the middle ear. The problem, though, there are problems that go forward with that. So the stakeholders here all have problems. First off, there's low reimbursement to the ASC. The ASC gets paid about $500 for this. And from start to start, meaning from the start of one procedure to the start of the next procedure, it's probably 30 minutes. So at best, these procedures are break even for an ASC, and in many instances, they're probably losing money. The surgeon is subject to those productivity factors and is spending a great deal of time where they're actually not able to be seeing patients. They're waiting for the room to turn. They're responsible for the people to come in and take out the trash from the previous procedure, to environmental services to come in and mop and wipe down, et cetera. The patient and parent, if you've ever seen this done or if you've ever had a child done, it's very traumatic for both. The patient is basically torn from the arms of the parent and nobody's happy about it. And finally, the payer hates these. So, because even though they've beaten reimbursement down to the ground, just the sheer volume of the procedures makes the expenditures significant. That's why, in fact, one of the reasons why they will have you keep the child on antibiotics, because it's dirt cheap and they don't have to worry about the expense associated with a surgery. So the obvious solution is to move this into the office, right? So just create an office-based BMT procedure. But that's not possible. The tympanic membrane cannot be anesthetized with the local anesthesia, and the patient is non-compliant, so you need anesthesia. An Israeli company from Yochniam named Luminous tried an office-based CO2 laser treatment, and it was ineffective for two reasons. Number one, the pediatric immune system is so incredibly prolific, it would close in matters of days. In fact, in a young child, you could perhaps do a myringotomy at 8 o'clock on Monday morning, and by Wednesday morning, the myringotomy is closed. The second reason was the wavelength. They were using a CO2 laser, which can't be coupled to a fiber optic. It requires a knuckled arm delivery or a knuckled arm delivery system. As a result of that, you could only really treat one ear per treatment session, because you get that first ear, but after that, forget it. There was no way that in a child you were going to get that second ear done. So here you see how the myringotomy is performed. You make a small incision in the eardrum and you insert the tube. And I want to go back to something I mentioned earlier. The purpose of this surgery is not to place the tube. The purpose of the surgery is to get that middle ear ventilated, get all that fluid that you saw behind the inflamed ear out and allow that to dry up and allow the infection to subside. So mitomycin has been demonstrated to be effective in maintaining patency of a myringotomy for up to 16 weeks. And you see at the bottom numerous references that show that this can be done. And this is with a blade myringotomy. It'll stay open for 16 weeks. The problem, though, is not the tube. The problem is the OR time. So one day, I always used to teach things to students at my daughter's grade schools. And the second graders got electricity, and the gee whiz moment on that is I'd run a bunch of RF through my body and make a light bulb light up in my hand. And the sixth graders got a little better, more comprehensive understanding of what the electromagnetic spectrum comprised, how it was absorbed, how it was transmitted, etc. And one of the things that I would do is I would use an infrared laser, which is what we have here. This is a laser made by Iridex. This is really old, maybe 10 or 15 years old, and it's designed for ophthalmic use. And it emits at 810 nanometers, so it's infrared. This is a white balloon, inside of which is a black balloon. And what I would do for the children is I would ask, if I did it properly and Socratically, the children would invariably, one or two in the class would say, what's going to happen when I fire this laser at this balloon? And they'd say, it's going to pass through the white balloon and make the black balloon blow up. Thus, okay. Unfortunately, I just popped both. Sorry. Isn't that great when things go wrong?
We can all hear though.
Okay. What happens is that I should have inflated the black balloon more. The black balloon will blow up inside of the white balloon. And that makes sense, because if you take this white balloon, nothing happens. But if I take the remnants of the black balloon, or here, this is just a twist tie, you'll see it's beginning to smoke, right? So the black is absorbing the energy, the white will not. And that's the key. When I did that, and the black balloon blew up inside of the white balloon, I just had an epiphany. Stain the mitomycin. Why not just stain the mitomycin? That would solve the problem in total. And you should stain it such that it will be dark enough. There's the remnant of the black balloon, and again, you can see it smoking and burning, but the white balloon will not. So what's the whole point? You want to be able to stain the mitomycin such that it will absorb the energy. So SPDM is born. So what do we do in terms of picking up that stain? Well, this laser emits at 810 nanometers. And there happens to be a drug, FDA approved, on the market. It's an angiography stain called ICG, indocyanine green. And this actually is part of an asset called Acorn, which is currently in reorganization. And it's possible that this asset itself could actually be pulled out during the time of reorganization. The peak, this has peak chromatic uptake at 808 nanometers. Here you can see the 800 nanometer line. You get close to 810. It's a perfect match. The second thing you need is an antimetabolite. You need mitomycin. And that's what we do. This is mitomycin. This is mitazole. And it's important to note that this is 0.2 milligrams per vial. There are many people that would tell you that's impossible. You cannot get a fill that small because the nominal fill volume is less than the tolerance of the filling equipment. But in fact, that's kind of our secret sauce. We figured out how to solve that problem. The other thing I'll point out to you is that within the mitazole kit, which is approved for ophthalmology, you'll see some devices that are made by ICU Medical. Mitazole, mitomycin C, is a NIOSH recognized hazardous drug. And as a result of that, when I put the kit together, when I invented it, it's a hazardous drug. It was just common sense to me. We need to control this. Well, as it turns out now, the United States Pharmacopoeia has established a new chapter called USP 800, and these sort of devices are required. And we have used the ICU Medical products from the start, and we'll continue to use the ICU Medical products. The last thing you need is an 810 nanometer laser. And this is provided right here. And there are many off-the-shelf versions. Here's two that are common, but it's very easily accomplished. So to make this work, you need indocyanine green, and you need a syringe to withdraw the ICG. All right? Then, for the sake of convenience, I've already attached these devices. You will take the ICG, connect it to the mitomycin, take to dissolve, withdraw it back into the syringe, and now you have 1 ml of mitomycin C stained with ICG. One of the things that people that are familiar with the approval process that cannot be overemphasized is that this is a drug device combination product, but CDER's review, one of the huge parts of this is module 3, chemistry, manufacturing, and controls. This is an approved drug on the market. Module 3 is done. This is an approved product. Module 3 is done. Process and method development are complete. Process and method validation are complete. All of the engineering batches, the validation batches, the processes associated with manufacturing are complete. We supply this. Our supply chain is already established, and a huge part of the regulatory submission is complete. So what do we have to do here? We have to do an experiment, because just because you have a balloon blowing up inside of a balloon, or just because it burns black and doesn't burn white, what does that mean? So we did an experiment in a temporal bone lab, and we used an 810 an NIR laser. For what it's worth, we used this the laser from Angiodynamics, and we have a cadaver temporal bone, and down here you'll see the tympanic membrane. So we would stain the tympanic membrane, and what we wanted to find out is how much time is it going to take to get that, to create a fenestration in the tympanic membrane? How much power is required to create that fistula? We wanted to see how selective the energy absorption was. And finally, we really needed to know that once that tympanostomy had been created, what's going on beyond the tympanostomy in the middle ear and inner ear. A good example about the time is a great example is that if it took seconds to happen, you're not going to have a compliant patient. So the results of the experiment provided the proof of concept. It came out that a 0.1 second pulse, a tenth of a second pulse, 7 watts of delivered energy, provided a consistent 2 millimeter tympanostomy at the site of the surgeon's preference, and also in the shape of the surgeon's preference. So typically you want it to be in the inferior aspect of the TM. Gravity is not just a good idea, it's a law. What's more, if you could make it in a half moon, you're going to have something much better than just a round fistula. You're going to have something that actually conforms to the anatomy of the child. We saw that the energy was absorbed solely at the site of the stain, and that was critical. Just as I said that you saw that the white would not take up the energy and the black would, we saw that the area that was untreated, the energy ignored it, and it ignored the energy. It was not within its portion of the spectrum for uptake. So at that point, we then excised the TM and did visual examination of the middle ear and the inner ear under microscope, and it seemed to be in perfectly pristine condition. It suggests that the brevity of the pulse and the limitation of the energy absorption limited the myringotomy and limited the photoablation to the tympanic membrane.
And if the tubes are meant to stay in for 6 months or a year to get past this infection point or the problems that it's causing, is this also a half-year fix, or do these drain holes stay open?
The first thing to point out is nobody wants a tube in for 6 to 12 months. what you're trying to do is you want to drain the middle ear, and you want to find relief of the infection. You want to be able to, that ventilation allows the infection to subside. So what you need, this is 16 weeks, it's 4 months before closure. That's on average. For tubes, they typically extrude spontaneously. So just as this will close spontaneously on its own, the tube is extruded spontaneously just by the healing process of the membrane.
Okay.
So what are the next steps? This company needs a CEO. I have managed this. I've had a great privilege in my career to be associated with developing therapies that have actually improved patient care. And that's why I have continued to fund this, and that's why I've kept this alive. Because I truly believe of all the things I've ever invented, this is the one that can have the greatest impact. So a CEO would need to submit a grant to NIDCD and do a controlled animal study to demonstrate safety and efficacy. You would do this in a chinchilla model, and you would do bilateral myringotomies. One ear would be a study ear, mitomycin C stained with ICG. The fellow ear would be done with just the ICG alone. And then you would track the efficacy based upon the length of patency. Prospective randomized mass controlled of a number sufficient to drive power statistically in the study. The results of that animal study, presuming success, would allow us to go back to the FDA and establish guidelines for phase 1 human safety. So the next thing the CEO is going to have to do is not only go back to get this to the FDA, manage the study, get to FDA, but then also assemble a team and to include a board of directors that can go out and raise some serious size Series A round of financing. So the CEO is also going to need to engage regulatory and industry. So we need to engage breakthrough. We need to gather breakthrough status with the FDA. It is a combination product, so you're going to have the review of both CDRH and CDER, but in an unofficial meeting with Malvina Edelman. She runs ophthalmology and ENT on the device side of FDA. He was very excited by this project, and she asked me if she could actually lead this through FDA. So we not only have a friend at NIH that wants this project to happen, we have a friend inside FDA that wants to see this project happen, gain breakthrough status, and ease the regulatory cycle. We need to re-engage corporate interests. Both Medtronic and Novartis expressed interest and actually wrote formal letters of support to NIDCD at the time in 2009 when we assembled the grant, when we received the grant. I could remain engaged PRN, and that's going to be determined by the operating team. But as a matter of fact, I'm not going to be part of that operating team. That's not really my decision. That'll be up to the new stewardship. So what are the benefits? And this is why I've stayed so close to this and want to see this happen. It checks every single box that needs to be checked by new products and new technologies coming in medicine for the future. Number one, it reduces costs because there's no time in the operating room. Number two, it reduces encounters with the system. Instead of going through chronic treatment with antibiotics, it's one single curative treatment and you're done. It reduces the use of antibiotics, and that is a looming hiding spot that is going to explode. I am certain that my grandchildren will live to see essentially the current range of antibiotics no longer be effective. It expands care. One of the things that NIDCD loved about this is they said you could do this in a neighborhood clinic or in a Walgreens or a CVS, and the answer is you probably could. It eliminates general anesthesia. We know that general anesthesia to children under the age of 5 results in cognitive deficit. If we can get that child off of antibiotics and do a curative treatment, one single treatment before the age of 5, we eliminate that entire risk associated with general anesthesia. This can also potentially move from the subspecialist. This is typically done by an otologist. So MD, transitional year, residency in otolaryngology, and then another 2 or 3 years fellowship in otology. This can move all the way down potentially to the pediatrician, the primary care. And that's an incredibly important move. All of these things are things that are supposed to be done by the Affordable Care Act. We check every single box. And we assume this is going to be preferred by patients, by parents, by providers, and by payers. So what remains in the near term, this needs to transition to new stewardship. It needs a CEO, and this is going to require a pretty sophisticated CEO. It's that simple. This is somebody that knows how to deal with NIH and writing grants, knows how to deal with FDA, knows how to raise money, has done it before, knows how to manage a board, and they're going to have strong familiarity with the regulatory cycle. They're going to have to engage FDA and build a team. Finally, they will need to still invent some devices that are for delivery of both the Mitomycin C and for the energy. The Mitomycin C can be delivered in several different ways that I've prototyped, and the energy would be delivered bilaterally, simultaneously, through conformal earbuds. Not terribly different than what you see here, where they would be put in to the patient's ear, a single shot, and it's done. And then they would have to engage serious venture capital money. So with that, I will open the floor if there are questions. And
George or Ed, is there current competition out there that's in this space?
There's not. The only competition that we saw previously, of course, was the Otolam from Luminous, and at this time that has been that's been removed from the market. Actually, they discontinued.
Okay.
Do you have any questions, Kelly, that are coming to you?
I want to make sure that all of our participants should be allowed to unmute themselves. Lisa, could you speak up just so I know that folks can, if they want to, ask a question? Got a question from Alan Clevens. Is the procedure painful for the child?
Great question, Alan. I would liken this to a child getting shot, and nobody is really going to want to do that. And you've taken your own children to the doctor to get their vaccinations. Nobody likes that, and it's one of the things that I think the best way to address this, the best way that I would envision this being done in the clinic, is that the child is sitting in mom's lap, and the doctor comes in. The delivery of the Mitomycin C would be accomplished through a modified otoscope, and a child that's had ear problems has had the otoscope put into the ear many more times than one. The way that this is envisioned is, honey, let me look at sitting child in mom's lap. Let me look in your ear. Mitomycin is applied. Let me look in your other ear. The mitomycin is applied. Direct visualization would be facilitated through this bifurcated bud. They would apply it to the ear, and it's a 1/10 of a second burst. It would be like getting a shot.
Is there any risk of healing to the ear that's been identified to date?
Well, we'll find that out in the animal study, won't we? I think there was one paper published out of Italy some years ago with a guy using an 810 laser with a contact laser fiber. So they were just heating up a sapphire tip, and they did not have good results. There's any number of reasons to speculate. However, the overwhelming majority of the literature contradicts just that. So that's why you do the animal study, right?
Okay. Is the company separate from Mobius Therapeutics?
Yes, it is. It was spun out as a condition of the seed round funding. The people that funded the seed round received pro rata shares in Mobius Therapeutics and Mobius Otologics. Since that time, I funded this, my wife and I have funded this on our own. And at this time, we control roughly 70% of the shares.
I have a question.
Hi, Dr. Schwartz.
Hi, how are you? Enjoyed the talk. With those earbuds, how were you able to aim the laser in the proper quadrant where you wanted, inferior, as you said?
It would be done under direct visualization, Dr. Schwartz. So you would have a CCD that would provide the visualization, and you would have an LED to provide the illumination. And by adjusting the monitor above the child's head, behind the child, they will then be looking at that. It's not different from, not terribly different. You've seen the Alcon virtual reality systems where they're using digital imaging. It'd be very similar to that. They would line up the laser when the aiming beam is at the proper site, fire.
And you can do that through those earbuds, I assume.
Yes, sir. There'd be the earbuds would involve one fiber, which would be a laser delivery fiber that's quartz, and also a CCD and an LED. Few people know this, but St. Louis makes most of the laser fibers in the United States.
Okay, thank you.
Hey, Ed, Dennis.
Hey.
Dennis Radnauer here. Question for you. My wife's a PA, so she had armed me. PA in ENT, so she had armed me with a couple of questions that she wanted clarification on. What is the lifespan of the surgery? I think you alluded to a little bit, but I didn't maybe hear. What's the lifespan? How long does this last? And how does that compare to tubes?
The literature demonstrates at this time a mean, if you take all 5 of those papers that I showed there, 16.2 weeks. 4 months.
4 months, okay. So so they would need to come back in then if they wanted it to last as long as tubes, because I think, what are tubes, a year or something? They would just need to come back in?
They would. It's you could do retreatment. It's also a case of if you can keep the child off of antibiotics, if they or minimize the use of antibiotics. 4 months, very often you find this child back in a week or so as the tubes extrude, and they're back under another general anesthesia. We feel the risk is definitely worth the reward.
Great, thank you.
Bill, do you have a question?
On behalf of Missouri Biotechnology Association, our board of directors, many of which are on this call, we thank you, Ed, for bringing this program to us, bringing this opportunity. I think that opportunity to share the deep promise that's there, that opportunity for really improving patient care and access, and doing a novel, game-changing business from what you've started, and your request to get the word out that we need a strong CEO coming on to take it to the next level. Really, that's great news, and our folks will be happy to spread that message. We'll make sure most everyone in the invitation got Ed's direct contact information. So please follow up with Ed, and let's continue to make progress in this regard. Any other comments for the good of the cause before we close up? We're right on time.
I want to thank Mobio and everybody that joined. I was supposed to leave on vacation yesterday, but I actually have my bag packed and I'm getting ready to get into the car and go. I'm going to be out of pocket for a few days, but back in the office the beginning of next week. Please feel free, if you have questions, fire away, but understand that 2020 has been a rough year. And I'm going to take a few days to try and get some time on the water. So thank you all for joining. Send the questions along, and if you have anything else that you'd like to add, please feel free. Thanks, Kelly. Appreciate it.
Back to the science, people. Let's go.